again. Tēnā koutou katoa ko Jacqueline ko ingoa ngā mihi nui kia koto. So, greetings from New Zealand. We're exactly 12 hours behind, so it's in the later evening here, so I'm sorry I probably won't stay with you all that long. Anyway, I have been working on Vespula species in New Zealand for about 30 years now, and so I think we probably have learned a few things about invasive Vespidae that may be of use to you. So I'm going to give you a little bit of a taste of some of the work we've done over those decades, but focusing particularly on the more recent work. So one of the key things that's worth noting about New Zealand is that ecologically and evolutionarily we're quite distinct. So we've been isolated from the rest of the world for 80 odd million years, which means we have a very unusual biota. We have no native social wasps or bees and only 11 species of ant. But we do now have 2 species of Vespula wasp that have become invasive, and we have 3 species of paper wasp. So we're gradually accumulating more and more social wasps. And of course, honeybees have been introduced here for a while. Okay, now I've lost. Why can't I see where I click to move on now? That is weird. Gotta be a way. Sorry, I've lost the little button at the bottom to advance the slides. Any suggestions? It's just ridiculous. Ah, there we go. Now I can see. Thank you. Okay, so this was a bit of work that we did with a team of people from around the world back and published in 2011. But it was useful just to take a look across the globe as to how this family of Vespidae are moving around the globe. And I guess the key thing to note here is these things have been moved around the globe ever since humans have been trading, really, and they're still continuing to be spread. And you folk now with Vespa velutina certainly experienced that in that. And of course, since it arrived in France, we could add now a whole lot of countries that the hornet has spread to in Europe. So it's a moving feast, and the prediction, of course, based on that graph, is that these things will keep getting spread around unless we get much better at biosecurity than what we are now. Out of that work, it really highlighted these 2 Vespula species as being the 2 super invaders, if you like. So the 2 species that were turning up again and again in a number of countries. And again, probably things have moved along a bit. I'd probably count Vespa velutina in that now as a species that's gone on to invade a lot of countries. But anyway, both those 2 species, the Germanica and vulgaris, are 2 that we happen to have ended up with in New Zealand. And so a lot of my talk will primarily focus on those 2 species. So both of those species come from Europe, and you can see for Germanica the native range there in green and the red invaded range in red, including New Zealand, which is virtually smothered in red dots, but more recent invasions into places like Argentina and Chile. So these things are getting around us, I guess the key point of that. So some more recent work by a group of us led by my PhD student, Julia Schmuck, use microsatellite markers to look at a whole lot of specimens of both the Germanica and vulgaris from New Zealand and also in Europe. So that allowed us to, we were fairly certain that we could pinpoint that Germanica had come from the UK, not quite so sure where vulgaris arrived from in New Zealand because we had so few samples from Europe. But more importantly with that work, what we were really looking for was to see how much genetic differentiation there was in the species now that they are in New Zealand. And what it showed to us is that these things have gone through a massive genetic bottleneck, which isn't really all that surprising. But it means that because of that reduction in allelic richness, gene diversity, and the effective population size, it means that it's very difficult for us to, within New Zealand, track how these things are spreading and how mobile they are, because you run them through the molecular diagnostics and they all come out more or less the same. So this was looking again at the genetic diversity in these species, and again focusing on the German and the common wasp on the right. And what we looked at was the number of haplotypes that have been observed in those two species in different parts of the world. So you can see, not surprisingly, in Europe, where these species come from, there's a lot more genetic diversity, but in the invasive range, it's a proxy for the number of successful introductions. And so for many of those countries, particularly with Germanica, you can see that the number of individuals that arrived into these countries was probably really low. So perhaps in New Zealand, only two individuals, two introductions that have set off a whole cascade of events. It's a bit higher in vulgaris, and the steepness of those curves suggests we didn't actually get to grips with all the diversity there. But nevertheless, that sort of information is kind of, again, useful to us to help us assess what the ongoing biosecurity risk of these things is. They're getting around all the time. So this graphic is really just to help you get your heads around the differences between northern and some southern hemisphere. So not only are we 12 hours different from you, but obviously we're also 6 months seasonally different from you. So whereas in the northern hemisphere, the peak of the wasp season is happening in your late summer, August, September. In New Zealand, the peak for us is February, March. Also of note is that the length of the season is much longer in New Zealand because we're a much more temperate region than Northern England, where that other data is for. So these differences are useful for getting our heads around what the populations are doing. But one of the things that we did earlier on was develop a population model for Vespula in New Zealand, and we did it very early in the piece, which means there was a huge number of gaps in what we didn't know about the biology and population dynamics of these species, but allowed us to pinpoint which were the critical gaps that we needed data for. So that population modelling was used partly as a predictive tool, but also as a way of identifying which were the bits that we needed to focus our research effort on at the beginning. And then later we were able to use that to assess what the targets were for control and where the likely weaknesses in the biology were that we could use, where in the population cycle should we be targeting control. So more on control at the end of this, but what I wanted just to look at for the moment is a little bit more about the actual invasive, what might be driving these invasions. So one of the ideas was that when they arrived into New Zealand, the reason that they flourished so well was because they didn't bring all their natural enemies with them, particularly the pathogens. And but some more recent work done by Phil Lester and his team just ruled that out, really. So these two species in New Zealand seem to have just as many pathogens as was found in the native range. And there also seems to be a bit of a reservoir in some of those pathogens with other social insects, particularly the honeybees. More recently, we've also looked on our small offshore islands in the northern part of New Zealand just to see what might be helping drive those abundances and distributions on islands. And the first surprise really was that many of these islands are small uninhabited islands, but they were just riddled with social wasps, Vespula and particularly Pilistes, actually. But it would seem that the key things that were driving the abundance of Vespula on those islands was the canopy cover. So the more forest that there was, intact forest, the less likely it was to have high densities of Vespula. And obviously, the further offshore these islands were, then that was also led to fewer wasps. And again, probably not at all surprisingly, it was also positively correlated with human settlement. So the more people were going to and from islands, the higher the density of Vespula wasps. Okay, so I'm mostly going to focus on the ecological damage, but I just also wanted to touch on the economic damage because one of the things we always struggled with in gaining funding and support for our work was trying to quantify what the actual economic damage was. And actually fairly late in the piece, some work was done by some economists to try and put some numbers on that. And I'm not entirely confident how robust some of them were, but it gives a ballpark of where the main economic costs for New Zealand are. And perhaps not surprisingly, given one of the main economic earners in New Zealand is through pastoral agriculture. So that turned out to be really high cost there. And it was particularly around the additional fertiliser and clover seed that needed to be added into those systems because the Vespula were interrupting the pollination of clover by disrupting the other insects that would normally be doing that pollination service. And then the next highest one by far was the indirect effect on apiculture, and that was primarily because of the loss of honeydew due to wasps feeding on that. And I'll talk more about honeydew in a second and explain that was. And then there were direct effects on the bees themselves by destroying colonies or killing the bees directly, and then associated other effects. Anyway, 130 million would sound probably like nothing in Europe, but New Zealand's a much smaller economy, and it is relatively significant for us. Okay, so I'm going to whiz through the ecological impact. So I said I'd come back to honeydew. So honeydew is this sugary exudate that's found in endemic scale insects in New Zealand. And in about 1 million hectares of our forest, we have these scale insects that virtually coat the trees in these sugary drops. So New Zealand has, in some of our forests, we essentially have created, we have a wasp paradise of sugar-coated trees. And it is in those forests that we have the really insanely high densities of wasps. So enough wasps that, the forests at certain times of the year are closed to the public because it just becomes too dangerous to let people into some of those walking tracks. Anyway, the ecological side of that is the wasps are feeding on the sugar and they're removing vast quantities of it and actually reducing the abundance to just a fraction of what would normally be there during the peak of the wasp season. And that's a real problem because many of our native birds and insects and lizards would all normally be feeding on that abundant sugar resource. So that is a major ecological impact. But wasps, of course, don't just feed on sugar. They also feed on a whole range of other things for their protein. And so a lot of work has gone into trying to quantify that. And so a series of experiments looking at the predation rates on caterpillars and all-web spiders came up with the estimate that we really need to, if we want to conserve those more vulnerable invertebrates, then we need to reduce wasp abundance in those New Zealand honeydew forests by about 90 to 95%, which is huge. But it was actually really useful to have those targets kind of quantified really early on because it it gave us something to go to funders with and say, well, it's no use trying to, just kill a few wasp nests here and there. It's not going to cut it with the amount of damage that they're doing. We need, we're likely going to need multiple control tools and we're going to need really effective ones. They're not just eating caterpillars and spiders, though, of course, wasps are eating all sorts of things. And this was another piece of work done by the same PhD student, Julia Schmuck. And in this case, we used DNA meta-barcoding. And this was a really new technique that was developed in collaboration with some other colleagues. And it was very cool because what we were able to do was take the excrement that the wasps deposit into the cells and extract the DNA from that, which means rather than the more detailed work that we'd done years ago of catching the wasps as they're coming back to the nest, grab their food off them, try and identify it, very laborious work that is not usually very successful because it's pretty hard to do diagnostics when you're just getting fragments of diet coming back in. So this technique allowed us to get a really good look at what they were eating across the season because, of course, what they're feeding to the larvae is accumulating in feces across the season in the bottom of the nest. So that seemed to me really cool way to be able to get it a more detailed look at what they were eating. And in this case, Julia was able to compare all 4 species of these introduced wasps occurred on a single island. So we were able to look on one island in detail and look at how they were partitioning the resources. And we lost a bit of time. I'm going to skip this one just to make up a little bit of time because I wanted, again, to come back to this honeydew. So we have these trees that are coated in honeydew, and every time it rains then the honeydew would wash off the trees and get added to the forest floor. So, of course, if wasps are removing that honeydew for 3 to 4 months of the year, that makes a huge difference in the amount of carbon that's being added to the forest floor. So it was actually a 4-year field experiment. We were adding honeydew at different rates, and that allowed us to get at the sort of question of how might wasps be impacting the soil food web and the actual nutrient cycling. So if you have lots of wasps in the forest, it actually slightly counterintuitively increased the litter decomposition. And the reason for that is because the wasps are removing the sugar from the soil around the trees, and the sugar would normally bind up the nutrients so that it slowed the whole decomposition rate. So if you have the wasps in the system and they're removing the honeydew, then that actually increased the litter decomposition. Anyway, the net answer was that, wasps are having quite a significant impact on that soil food web, and increasing the carbon sequestration by quite a significant amount. So this just sums up the type of impacts that those Vespula wasps are having, and they're affecting the ecology at a whole lot of different levels. Some of the things, the birds and the beech trees with question marks, I strongly suspect if we'd be able to get a handle on those, we would find there were strong effects there. But everything else that we measured, we found that they were having, quite significant impacts. And in fact, for wasps would be considered our most serious insect pest by a long way in those natural systems. Okay, so I just want to finish off by, again, fairly quickly covering the range of control tools that we've tried. Firstly, the poison baiting is by far the most successful thing that we've done. So this relies on the fact that wasp adults can't digest the protein by themselves. They have to take the protein back to the nest, feed it to the larvae. Larvae have the digestive enzymes, and therefore, and then they, share it around the rest of the colony. So if you can get a poison into your protein bait, then they'll take it back to the nest, and voila, you kill the queen. Nice in theory. Eventually, we got this sussed in terms of fipronil, and we found that was really pretty successful, so that in areas that we were using it, and these were fairly large areas, 110 hectare blocks, that was able to kill about 90% of the colonies. I'm not then going to go into the story of why it then took so long to get this product registered commercially in New Zealand, but eventually we did, and it is now available commercially in New Zealand. And it's a private company now that markets this. I have nothing to do with it anymore, but it's actually been surprising to see how much uptake this has had around New Zealand because it works, and people have been desperate to clear wasps out of their blocks of land for various reasons. So poison baiting, awesome. If you can find the right toxin that works in the right formulation and get through all the processes required to register a product. And that's just a little bit of data that's showing that if you, in areas where they had treated with this fipronil poison bait and reduced the wasp numbers, and then the honeydew resource bounced back in comparison to areas where they didn't do treatment. So pretty effective, at least on a relatively small scale. We've also spent a lot of years trying to get biocontrol to work. Excuse me. So I'll deal with the bottom one first, Vespula vesparum bara, which was a biocontrol agent. It's an ichneummonid parasitoid that was imported from the USA, and despite a huge amount of work to get it through the system and reared up and released, it didn't establish. But the other closely related species that was brought in, Vespula vesparum, this came from Europe. It did establish, and initially the results were looking really promising. We were getting relatively high infestation rates per infested colony. So you can see here back in the late 80s, when it first established, we were getting almost 800 parasitoids per parasitized nest. But then it dropped off dramatically. So the parasitoid was spreading and the number of nests infested was going up, at least to some extent. But the number of parasitoids per nest just dropped right away until, we're almost back at zero. And in fact, it was only because we had some really good folk that were really good at spotting these things that we were able to detect it at all. So we never quite got to grips with what was going on there, but for whatever reason, this thing came in, established, looked as though it was doing quite well, and then just didn't cut it at all. The net effect, not surprisingly, when we had, this is the one site where it had established, and we were following the density of wasp nests per hectare per year. And then the other 5 sites are all sites where the parasitoid doesn't establish. And so don't do your head in trying to look for the pattern. There is no pattern. There was no real difference in the population trajectory at the parasitized site compared to the other sites. So huge amount of work, and the net conclusion was that didn't work. I'm not going to go into any of these novel control technologies really, because A, it's not my work, and B, we don't have a lot of time. But just to point out to you that there's been a bit of a renaissance of effort looking at a whole lot of potentially novel control technologies in the last few years in New Zealand. So there are people in New Zealand that have looked at these. Most of these have been ruled out for the moment as not getting us anywhere particularly, but there's still some work looking at the potential of a gene drive. All right, so like to just finish up quickly by saying that these Vespula wasps in New Zealand have turned out to be really ecologically damaging. And I think, there's a whole range of factors that have come into play there. But one of the biggest drivers is just the ecological context of New Zealand, that we had that honeydew resource that was just an amazing sugar resource to drive super high abundances, and obviously not much in the way of predators or competitors, because we had a system that had evolved without social insects essentially. The control is extremely difficult, time consuming, costly to develop these things, but the toxic baiting has proved pretty successful, although of course the use of toxins over large areas year after year after year is probably not going to be a good thing in the long run. So we definitely need to keep looking at new environmental friends. If I had my say as to how we would also invest the money, we would look at new tools for detecting the things, invest in our border biosecurity, because the last thing we need is to add more species to the mix. And yeah, I'm not at all keen on ending up with Vespa over here. Thanks, you can keep those in Europe. So to the huge number of people that have involved in this research over the decades, and that's by no means all the people that have been involved. They're just some of the ones that were involved in the work that I've mentioned. But this has been, decades and decades of lots of people's work, huge amounts of effort, and we've cracked it all. So thanks for listening, and hopefully we've got a few moments still where we can try for some questions.